So there are some plants that have evolved solutions to the challenges of photosynthesis. And the first group of plants we're going to look at do something called C4 photosynthesis. And here's the figure in the book that ties into the things I'm going to draw here. Now for C4 photosynthesis, there is both a biochemical solution that we'll look at in a second, but there's also an anatomical solution as well. And so we're going to draw again a picture of a leaf, giving ourselves lots of space here. We're going to start with the epidermis, and then again here's the lower surface of the leaf with these stomata. By the way, I'm drawing this with the stomata on the underside of the leaf. That is most common, but there are certainly leaves that have stomata on the upper side and leaves that have stomata on both sides. But we're going to start off with this waxy layer of cells called the epidermis here. Okay, now inside here we still have mesophyll cells, but they're actually surrounded by another circle of cells. So I'm going to start here and draw 3 circles. Now the innermost circle of cells, I'm going to label this with a V, and that V stands for vascular cells. So we'll learn more about the xylem and the phloem, the cells that help move water and sugars around inside the plant, but that's what's in the center of this circle is the vascular tissue. And now what we have going out from there are these concentric rings of cells. And you can see there's an outer layer of cells and an inner layer of cells. The outer layer of cells is called the mesophyll cells, same term as before. And then we have this inner layer of cells, and these are called the bundle sheath cells. All right, so there's a layer of mesophyll cells surrounding this layer of bundle sheath cells, and there's still airspace here in the leaf, to allow for gas exchange, and that's also a potential way to lose water as well. Now, this special kind of anatomy that we see here in these C4 plants is called Crohn's anatomy. Those 2 concentric layers of cells. I always assumed that was named after a person until I looked into it a little bit more and found out that it was actually named after this sort of ring-shaped cake called a Kranz. This is a Frankfurter Kranz that you can get if you go to a bakery in Germany. But it's got this sort of ring here. Now here's a microscope picture where you can actually see these rings of cells here inside the leaf. There's the bundle sheath cells on the outside, or the mesophyll cells on the outside, and then the smaller bundle sheath cells on the inside. All right. So this is a really, really important figure and one that you have to understand really well if you want to be prepared for exams in this class. Because what we're going to do here is side by side compare and contrast these 2 solutions for photosynthesis that we have. And over here, this is going to be our diagram of C4 photosynthesis. Okay, so what's shown already in this picture, actually I'm just going to zoom in on this side here. So here's the C4 part. And by the way, when you're taking notes on here, save this part here in the middle, because when we're all finished, we're going to label some stuff in there. Okay, so here's our C4 plant, and the upper cell here in our diagram represents the mesophyll cell. And the lower cell here represents the bundle sheath cell. All right, now you can see what's drawn in here, I guess, are really the cell walls of these 2 cells. And you can see that there are these large openings here between them. And basically, those are called plasmodesmata. I should label that. Plasmodesmata, a good vocabulary word to know. And this is pretty common in plant cells where the cytoplasm of these cells are linked through these large openings. So basically they are sharing a cytoplasm. Okay, now the upper cell here, the mesophyll cell, is the one that's exposed to the air. So up here there's going to be carbon dioxide in this leaf airspace and also oxygen in this leaf airspace. All right, so we're going to start off with a 3-carbon molecule up here in the mesophyll cell called phosphoenol pyruvate, PEP. And this is a 3-carbon molecule. Whoops. C3. And there's an enzyme which takes this PEP carboxylase and also uses carbon dioxide as a substrate, and it converts it into a 4-carbon molecule, C4, and that's where the name C4 photosynthesis comes from, is from the synthesis of this 4-carbon molecule here. Now, I don't have room to write the whole name of the enzyme in here, but I just want to have a placeholder for it. This is called PEP carboxylase, so I'm just going to call it PEP-ACE for right now. But this is important. It is selective for CO2. So it doesn't matter how much of this oxygen is up here in the airspace, PEP carboxylase selectively pulls out the CO2 and adds it to PEP to make this C4 molecule. Okay, so what happens next? The C4 molecule is going to diffuse through the plasmodesmata and into an organelle here in the bundle sheath cells. And you might be thinking, hey, it's an organelle, it's probably the chloroplast. We're thinking about photosynthesis here, but no, in fact, this is the mitochondria. Sorry, I didn't mean to fake you out by drawing that inner membrane there. This is the mitochondria. And this C4 molecule is going to go into the mitochondria, and there another enzymatic reaction results in this C4 molecule being broken down back into a C3 molecule, and also CO2 is released. And that CO2 then, it goes into the chloroplast, which we can draw down here. And this is where the Calvin cycle is taking place and where photosynthesis is taking place. Now, through a series of steps, this C3 molecule here that's made is going to be converted back into the starting molecule, the PEP carboxylase. So what we have going on here is this way of selectively pulling in CO2 into the cell through this first enzymatic reaction, and then down here in the bundle sheath cells, that CO2 is then released to be available to the Calvin cycle. So it's important to notice what we have up here is we have carbon fixation taking place in the mesophyll cell, and we have the Calvin cycle taking place down here in the bundle sheath cells. And this is an important concept, and you'll see this in the textbook, talk about spatial separation. Sorry, we got a lot of stuff labeled over here. So spatial separation, it's sort of saying that, all right, carbon fixation and the Calvin cycle are taking place in these two distinct spaces. And this is meaning that down here in the bundle sheath cells, where the Calvin cycle is really active, where Rubisco is really active, there is going to be higher concentrations of CO2 relative to oxygen. And that is going to result in less of a problem with photorespiration. So what does that mean? If we can reduce the problems of photorespiration, right? Then remember, photorespiration, that's where Rubisco uses oxygen instead of CO2 as a substrate. If we can reduce that problem and make photorespiration less of a thing, then this plant can spend more time with its stomata closed. It's okay if the oxygen levels build up in that airspace a little bit, because there's this method for selectively pulling out the CO2. And that's what this spatial separation of carbon fixation and the Calvin cycle achieve. All right. What's the end result? Now, you've seen this already. Remember, to make that 1 gram of cellulose, 1 gram of fixed carbon, a C3 plant is going to use about 400 milliliters of water. So in a C4 plant, that amount of water is a lot less, right? This is just 250 milliliters of water to make that same 1 gram of carbon. Now, we're still, 1 gram of cellulose, right? So we still are losing water for sure, right? We can't eliminate water loss entirely, but because these plants can spend more time with their stomata closed, they are going to lose less water to the environment. Now, some examples of C4 plants. A lot of grasses are C4 plants, like these corn plants here, and this is sugarcane down in the corner. And these are plants that are able to live in slightly more dry and arid conditions than a lot of C3 plants. And so this is a pretty common adaptation in the grasses.